Так, добре. Всіх вітаю. Сьогодні ми зробили це в форматі, запросили Юрія у форматі квантового семінару, але це у нас такий семінар з одного боку. Зараз зрозуміло, що він міждисциплінарний і присвячений методу, так? Але також хотілося б, як формат нашого квантового семінару, сюди застосувати у тому плані, що ми лектору не робимо ніяких обмежень у часі, і слухачам не робимо обмежень у питаннях. І Юрій погодився, і навіть, можна сказати, був ініціатором такої зустрічі, тому що ми йому вдячні за це. Юрій, я тут трошки, я бачив на сайті, про нього є CV, так, і ми трошки говорили, що він взагалі родом з Одеси, але він вже дуже давно вже працює за кордоном в різних місцях, і головне, що зараз він працює в принципал електронний мікроскопіст, так, головним електронним мікроскопістом в цей Bioimaging Center, який знаходиться при, він краще сам, звісно, не розкаже, який знаходиться при Даймонді синхротроні. І ідея в тому, що є зараз багато можливостей, насправді, на різних синхротронах, зокрема у них, якими українські науковці чомусь не користуються, тому що справді є можливості і віддалених експериментів, і тим, хто можуть їздити, просто експериментів вживу. Тому от Юрій про це розкаже. І the talk will be in English, I think. And anyway, someone else from Solaris, they said they will attend, but maybe anyway. Yuriy planned to do it in English.
Yes, я дуже вдячний за цю можливість розповісти про які є можливості на Даймонді. І так, з технічних причин я буду давати цю презентацію англійською, але якщо ви хочете задати питання українською, я можу відповісти на будь-які питання українською або англійською. Корекція, я сам родом з Миколаївської області, але я прожив в Одесі 9 або 10 років, тому що я закінчив Одеський університет і потім намагався закінчити аспірантуру, але прийшлося закінчувати вже в Нідерландах. Добре. So, the title of my talk is Research Opportunities at Diamond Light Source, X-rays and Beyond. So, Diamond Light Source is UK national synchrotron, which is located in southeast of the country. It's just south of Oxford. And it was designed and built as user facility to do synchrotron experiments. A lot of work went prior to planning the synchrotron. And synchrotrons quite often are viewed as engines for physical and material science. However, because of partnership of UK government with Wellcome Trust, which is private funded research in UK, and the emphasis of Wellcome Trust is on life science research, the life science, including like medical science and biology, was taken into account during the planning of synchrotron. Therefore, Diamond is one of the best planned synchrotron to do not just the physical science, but also a lot of biological applications. So where we are located? So we are located in just south of Oxford, as I said, and you can see Diamond Ring with all the facilities embedded around it, but also it's in the middle of a research campus, which has a lot of other facilities, which help to get the most from sophisticated instruments located at Diamond. In particular, what would be important for this talk is Rosen-Franklin Institute, is newly built institute, which the main purpose of which is to help physicists create new, as they say, disruptive methods to help biology and chemistry as methods which would make like step change in what we do and improve science in biology and chemistry. And also IBIC, IPSIC. IBIC is part of Diamond. IPSIC is partnership with industry and University of Oxford. IBIC and IPSIC are electron microscopy facilities. So this is goes with title, one part of my subtitle, like X-rays and beyond, because we not only do experiments with X-rays, we can also offer electrons and other possibilities here. Research complex at Harvard is pretty much a research hotel. So it is separate from Diamond, but Diamond is a stakeholder in the research complex, and it provides lab space for people to come from different universities and do experiments. Some people spend their entire like PhD or postdoc at research complex. Central laser facility is separate, but we have some collaborations. So some joint experiments are possible, although they need to be approved separately for Diamond and Central Laser Facility. ISIS neutron and muon source, we have joint proposal scheme. So potentially one can apply to do experiment both the Diamond light source and neutron and muon source facility. We have collaboration with a lot of Rutherford Appleton Laboratory. It's, I think, the oldest on site, and the whole site is actually called like Rutherford Appleton site. It has increased security because of nature of some experiments, but also it provides us access to high performance computing and some groups like scientific machine learning group, which is located in this building. And nearby, we also have Mary Lyons International Mouse Center for Functional Genomics, which can provide some mouse lines for us to do experiments on living organisms. Diamond has 33 beam lines, which are organized in 8, well, it's 33 X-ray beam lines, because microscopes also considered part of beamline business, but they qualified as non-synchrotron beamline. The beam lines and microscopes are grouped into 8 villages based on application. But the experiments can be easily made between different villages. So if people would like to access different instruments, there's no hard borders between these villages. The villages are macromolecular crystallography, which I'm going to touch on today, imaging and microscopy, biological cryo-imaging crystallography for material science and chemistry, structures and surfaces spectroscopy, magnetic materials, and soft condensed matter. The more information can be found on Diamond pages if you go to this annual review. So there are some papers which are published by users, and Diamond review and the highest impact papers normally end up in this review. So you can see what kind of science can be done on those beam lines. I think they group by group for different villages. You can also go back in time, because I think it's the log goes down back to 2017. So also more information can be found on the web page. If you go to instrument tab here, all the villages, and you can also look at specific instrument, but please look inside, because many things have more than one instrument, like EBIC, for example, or B24, which has both soft X-ray tomography, but also super resolution structured elimination cryo-electron microscopy, sorry, cryo light microscopy. In general, I must say that Diamond webpage is designed as a main way of interaction with user community. It's a bit convoluted, but pretty much most of the questions people have can be answered there. And please have a look, go through different tabs, check information there. It's quite big resource. What kind of experiments are performed at Diamond? That's again some extracts from light source, Diamond Light Source annual review. So you see that in the past couple of years. Biology and biomaterials is more than a quarter of research done in diamond. If you add medicine, that will possibly go close to 1/3 of all research done at diamond. But also there's a big chunk for chemistry, physics, technique development, some interdisciplinary research where it's hard to throw a border, environment engineering, information and communication technologies, food science, and also archaeology and cultural heritage. So for example, we have scanned some archaeological artifacts to discover evolution of hominids in diamond on some of the beam lines. The size of samples which can be studied on diamond is very large, so we can look inside an organization of molecules and then find, for example, like new antibiotics that's ligand like a molecular and between molecular atomic scale, and then using resources of EBIC, we can go up the biological macromolecules and see the whole molecule, its arrangement in cellular membrane, then how complexes are assembled. We can trace it inside the living cell. For example, here is an example of correlative microscopy. The gray represents X-ray, soft X-ray microscopy, and the red is super resolution light microscopy tracing filaments inside the cell. So you see actually the filaments in the cellular context and can excuse me, study life as it is. On other beam lines, larger objects can be studied. For example, here is the inner ear of a mouse studied an I-13 beam line. Also, some dynamic studies can be performed. Here we see distribution of osmium and bromine, which are used in some cancer treatment inside the cells, and this was dynamic experiments, so people can measure different stages of how the distribution changes based on other factors. In chemistry, it's a wide array of measurements you can make. It's you can study structure. You can also study the dynamics of different materials. Also, diamond constantly evolving. We invest in development of new techniques, and there is B16 experimental beam line, which people can use to conduct experiments which are not easily done on other beam lines, which are mostly purposed for to service user community, and they normally have range of tasks which are easily performed and optimized. The focus of this talk will be a little bit towards biological science, based on what we discussed with Sasha earlier. I have more slides on material science from Solaris Synchrotron. I think they should be available now on Solaris webpage. On demand, I can send some slides. But please do make inquiries either directly to leaders with beam lines, or you can forward it. We can also send inquiry to me, and I'll find people if you're interested in knowing more about experiments in certain beam line or planning experiment at certain beam line. I can try to help with that. So for biological studies, one of the biggest village is macromolecular crystallography. This is 60% of diamond users, and the beam line is highly optimized to do crystallography and obtain structures of biological macromolecules which can be crystallized. You see the crystals can be very different size, so therefore this village has beam lines which can help you to study this crystals of different dimensions. The beam can cover from 0.4 to 100 microns in size. It has also a wide range of energies. For some tricky crystals, like crystals of viruses, things need to be done really fast because they pretty much blow up under the beam. So the information should be collected before they're destroyed in the beam. So that is also available. The range of molecules is really wide. It could be anything from small peptide to, as I said already, like human viruses. There are also, I think it's IO3 beamline, which actually has containment measures. So samples of infectious diseases can be studied there. I think up to containment level 3, which is COVID, I think, was classified as containment level 3. There is Diamond invests heavily into automation and helping with the processing of the data. Many beamlines of macromolecular crystallography have on-the-fly processing. So the claim is that users often don't really need to do anything at home. They can just send samples and then get back data ready for interpretation. That's the macromolecular structure ready for interpretation. Some users I know still don't believe it and try themselves, but the claim is that in most cases there's nothing which can be improved after diamond processing. So in macromolecular crystallography, quite often it relies on existence of already solved structure to do phasing, because you only get amplitude information, and phases need to be computationally derived from the amplitude information. The method of molecular replacement relies on intensities from known protein and knowledge of crystal lattice parameters to actually try to get phasing. However, Diamond has also abilities to do phasing de novo. If known structure not available or not suitable, one can do experiment to get the phases experimentally. To summarize, this is macromolecular crystallography is high throughput. It's a family of high throughput beamlines. All of them have remote access, so all the samples needs to be sent, but people also can come if required. But please check how long your experiment will last, because it's highly robotics and high automation means that sometimes the whole data collection is actually 10 minutes. So if you need to spend like a few days to come up to Diamond and then have 10 minutes for data collection, maybe that's just to log in and watch things happen and control things rather than be on site. But yeah, there's also some experiment, like maybe experimental phasing might require on-site visit to check things. Another part of MX Village is crystallization facility and membrane protein laboratory. This is facilities for people to learn crystallization techniques and also do some experiments. And I'll touch on this later. This is some added value which Diamond has in the community by not only providing instruments to solve the structure, but also necessary equipment to prepare the experiment. And one of the highlights, recent highlights, is that VMXM beamline comes live. I think they are accepting commissioning proposals at the moment. And this beamline is optimized to look at very small crystals of about 1 micron in size. Can be also smaller, but that's not, yeah, it might be tricky, but 1 micron means that the sample can be put on 3 millimeter cryo-EM grid. and people can then decide, do they want to do X-ray crystallography or electron diffraction, which I'll touch on later on. The next village I would like to describe is imaging and microscopy village. So it has different type of instruments. Some instruments optimized for full field imaging with different spatial resolution. That's basically the X-ray one can get in the hospital, but because we also have all these tunable parameters, it can be done at different energies, and it can penetrate different size objects of different size. Also, there's possibility of tomography. And another type is scanning microscopy. Scanning microscopy would allow to not just get the structure, but also gather information about the sample. Diamond investing a lot of time and resources in coherent imaging and ptachography experiment when image can be reconstructed from diffraction data. That helps to make, to find, basically to have the same experiment and obtain very different type of information. How is it split between different beam lines? So you see here, we have some beam lines which are dedicated to spectroscopy experiment and multimodal mode. Some which are more dynamic, some which are imaging beam lines, but they also can provide dynamic, they have ability to carry a dynamic experiment in 3D and 4D, because there are some environmental cells available. That probably wouldn't apply to B24 though. And something is in between. So Diamond has quite unique diode beam line, which can do both imaging and spectroscopy at about 10 hertz speed, if I'm correct. I'm not sure about this number, but in principle, one can obtain the data simultaneously. I'll show type of data one can obtain with this beamline later.
Yuri, the question, the image from diffraction, you said, yeah, it is. It means that you have some phase retrieval algorithms yes. There included, and it is just after processing or just during experiment.
That's a very good question. It's computational method, obviously, but Diamond strives to do it pretty much on the fly. Mm-hmm. That's what we try to do for all beam lines. So
it is just implemented there in this this end station in analyzer.
Yes.
Or...
Yeah. So the idea is it might be like depends on experiment, but in general, the drive of Diamond is you should see what you're getting from your experiment straight away.
Mm-hmm.
Might not be always possible to highest resolution. Obviously, some computation take longer. Yeah, but in principle, that's the idea.
Yeah, this means that it's everything already included. This software is already. It's not the case that you need this task to somehow to contribute to this task. How this algorithm could be improved or something. It's already done.
There's always way to improve, right? So if you have a good idea how to improve things, I think people will be keen to listen and help and to see if that can be done. We research and development is hand in hand with actually experimentation.
But this software, it's devised in your lab, or it is just commercial?
We use both, but most software for this type of experiment is actually either devised with Diamond or it's been in like collaboration. So Diamond is an interesting place because we are officially a company, although we got 86% funding from government and only 14% from other fund, well contrast.
How many from government?
86% we funded from government, yes. And this is why now there's a lot of tension, because government says that because we fund you, you need to obey all the rules which we put like on salaries, etc. People are not quite happy with this. But we have, because we like a company, and about 14% what we do is also offered to industry. We pay some industrial rates, a fraction of industrial rates on commercial software. This is why we try to get either academic software, either software which is free for use, or develop something in-house.
Okay, thanks.
Because otherwise it's getting a bit expensive. Right, so the range of imaging, because of all these different beam lines and also microscopes, we pretty much cover the whole range, and we can go from structure of a protein to bigger structures. And I think this gap here should be removed, because right now I think electron microscopy and B24 can close the gap between EBIC, IPSIC, and GO8 and other beam lines. I think
Actually, if you use pointer, we do not see your pointer. So
okay, apologies. Do you see it now?
Yeah, now we see it.
Thank you. Yeah, so basically there is a gap here, but I think that's just old slide, because I think now we cover all the range. We can provide information from sub-angstrom to hundreds of micrometers and millimeters. The energy range is also quite wide, from electron volts to mega electron volts. Diamond has diamond is 3 giga-electron volt to synchrotron, so we can afford going to higher energies if required. I think the biggest, one of the beamline actually has ability to study objects up to 2 tons, so quite big objects for industrial development. So how all this can be exploited? This is an example of a study of tooth decay. People would like to know how we get carriers, and how what can be improved there for better treatment and maybe better artificial teeth. So this is a tooth. You see that it was sampled from different places, and then this is non-carious part, and you see it's smooth and nice. This is here you see a feed milled specimen.
What are these rectangles? It is where you scan or what?
The rectangle, I think it's scanning microscopy, and then I13 tomography. I13
no, I mean that it left some traces on the specimen, and why? It is because image was scanned from there, or?
So you mean?
No, I mean not. Yeah, this like.
Yes, this is a scanning of the tooth after all these places were taken out. Mm-hmm. Yeah, basically it's samples from a tooth. What you see, this big image here is a human tooth, mm-hmm. And these samples were taken from different places, then colored based on what structure you see there. Yeah, this is a really big, comprehensive paper here. If you're interested to know more details, it's amazing. There's much more than I can show here, probably talk and so on. Yeah, but you see, you can then mill it with focused ion beam, which we also have on site, to a specimen you can study with other microscope technique. And in this case, it was I13 beam line. So you can see that there is the structure in in body of lesion has this porous structure, meaning that possibly calcium is removed from there. And then you can use other beam lines to actually see, test your hypothesis and see what is inside. So you've seen already these rods at high resolution. Now this rod was sliced into thin lamellas, flat thin lamellas, and people use X-ray fluorescence to find where calcium is located, and then also this is reconstruction technique with topography, which we used for reconstructions from diffraction experiments. And then you can also see different crystalline arrangements inside the tooth, the way how these rods are arranged inside healthy and non-healthy parts. And the number of beam lines was involved, I-14 and I-08. Another example of experiment, this is actually goes across villages because B24 is in biological cryo-imaging, but that just shows that you're not limited to one particular village. If you need science, you can just, and you have a good case, you know what you would like to do, then you can do a complete experiment here, which could be sufficient to get a publication. So the cells, this is like cells, which cancer cells, they sometimes photoactive platinum is used. The platinum, which can be photoactivated, can be used for cancer treatment. So the idea of this experiment was to see, does it cause any damage? What happens exactly? Because in medicine, often the proof is based on does it work or not. It's not much about how it works and what are the consequences. And obviously, for developing techniques, it's important to know how things actually work. So the cancer cells, which was treated, was incubated on electron microscopy grid, which is a 3mm mesh. Normally, it's copper or gold. Often for cells, gold is used because it's non-toxic. And then in B24, you can do super resolution microscopy if there are fluorescence in your cell, you can see it, and then on top of it, one can do soft X-ray tomography, that reconstruction of cell environment. So you see here they were able to detect where platinum is accumulating and then point it precise position in the cell. And then with other techniques, they were able with X-ray fluorescence to locate platinum inside the cell, and with SANES spectra to actually see changes in state of this organoplatinum compound. And that was done on I-14 hard X-ray nanoprobe. Dynamic experiment. So there are also environment cell availables. So in this case, there's growth of crystals inside the cells, which is kept at 40 degrees. There's sealed cell with some flow through it, and it's been incubated, and we see that this white crystal growth appearing. Other type of experiments, scanning X-ray microscopy, and this is again the IO8 has unique ability to give wide range of energies, and people were scanning across these energies to actually find different elements inside the cell to understand how Alzheimer plaques are formed. Diet. I mentioned this before. This is one of the beam lines with unique capabilities of both tomography and imaging. This is non-biological sample, that zeolite. As I was told, a lot of water purification plants rely on zeolite extracted from American mine about a century ago, and stocks are running low. So people would like to understand what it actually is so they can produce it artificially. And in this case, this beam line was useful because you can have a picture, but you can also say, as you see the 3D model you're evolving, you can say that, okay, I would like to have diffraction measurement from that and that place to understand what it's actually made of. And here you see like two different types of material are present, one on in the surface and another one is inside the particle. So to complement all X-rays, you can get also access to IPSC, which is often performs experiments with different beamlines. IPSC has two gel microscopes. One is a 200 kilovolt microscope, which is optimized for spectroscopy and used to find composition of materials. And the second microscope is optimized for imaging and also has a wide range of tunable environments. And you can see here the high resolution imaging with of a graphene layer and other materials. The joint experiment with other beamlines is routine. There's also a feed milling instrument. Potentially one can apply for feed milling and then get access to other microscopes if you would like to study materials you milled. Christopher Allen, who is principal beamline scientist in IPSC, he's really keen to view proposal before the submission and sometimes help to phrase it that there is best chance to get our time allocation. Now we come to IBIC. This is place where I work. IBIC was established in 2013, so for quite a long time there was problem with access to high performance electron microscopes, and people started to think that it would be nice to have a facility which would operate like synchrotron beamlines, so one can apply for time and get access to latest and greatest. So IBIC is a national facility for electron microscopy. We grew over time, so it started with two Titan Cryos microscope, which is top of the range for biological science, a 300 kilovolt instrument, highly optimized for biological science. It cools specimen to liquid nitrogen temperature, and you basically the way it has automated refill, so you can do cryo-electron microscopy, and you don't need to think about topping up liquid nitrogen, which was a big issue in old days, because it connects to liquid nitrogen supply. Everything is automatic. Microscope will tell you if there is any issue, so you can rescue a specimen if there is a problem. But normally it just runs 24/7, and you can just exchange samples and collect your data. And there are also two 200 kilovolt microscopes, which we know initially it will, the first microscope belongs to IBIC, and it initially was thought as screening machine, but now as we have more intricate experiments, we start using it for user time as well. And also research complex glacios microscope, which is belongs to research complex, but placed at IBIC, and we help to manage it, and also it uses IBIC infrastructure. We have two feed milling machines for biological specimens. They had to be optimized. It has been a standard technique in material science for many years, but the problem is that we look at water which is vitrified at high speed, and it needs to be kept below minus 154 degrees Celsius. Otherwise, amorphous ice will undergo phase change into cubic ice and will not be suitable for research any longer. Well, basically, you still can get information from there, but people will question validity because when crystals grow, they tend to disrupt biological structures. Recently, we got like a cryoclam instrument, which allows to see fluorescence of frozen specimens. Techniques we employ, it's most of the time is single particle electron microscopy. It relies on the fact that in the biological macromolecules are the same, And if you have a purified specimen of biological macromolecules, you can choose either to try to crystallize it, but it's not always possible. So one can freeze it on electron microscopic rate and then use cryo-EM to get the structure here. So you see that's our proof of concept that our system works. So we obtained the structure to roughly 1.6 angstrom resolution. It's obviously in biological cryo-EM, the biggest problem is not the instrument itself, but flexibility and properties of biological matter. And so the ball is on averaging techniques. How do you get information out rather than on instruments? So we're quite pleased to see that, yes, our microscope can perform to this resolution with the right sample. And cryo-electron tomography relies on getting information from a single specimen, which cannot be found in multiple copies. So it's optimal for studying cells and some other objects which have irregular shapes. So averaging is not easy to do, but it is also useful for now people use it to get information about molecules in its native contact, which we'll touch later. EBIC was designed as a data collection facility. So we do not have space and resources to offer sample preparation on routine basis. So unless it's a collaboration experiment, so we would expect people to prepare cryo samples at home and then or elsewhere, and then send us already prepared samples in dry shippers. However, we do offer to users chameleon sample preparation. This is a new technique which was developed in the US by academics and then sold to SPT LabTech, the concept. So they now try to build equipment around it, and in collaboration with SPT LabTech and Rosalind Franklin Institute, we can offer to users ability to freeze samples. The benefits here is that the sample is frozen at very rapid rate. And for some samples, the biggest issue is preferred orientation, because particles tend to attach to air-water interface and be fixed in one conformation, which is not good for 3D reconstruction. The rapid rates of freezing on chameleon helps to avoid this, because there is just no time for particles to migrate to air-water interface. The drawback is it's quite costly technique, but it's not user's concern. So if it is approved, then people will get time on chameleon for free, and also free access to the microscope to check the result. Normally it will be 200 kilovolt screening instrument to see what is frozen, and then the same samples can be kept at EBIC if people would like to collect high resolution data on high throughput 300 kilovolt instrument until this time is allocated. Micro ID, or some people call it 3D ED. This is electron diffraction. It's a relatively new technique for biological specimens. It is very useful at this point for small molecules, for chemicals, because it can provide sub-angstrom resolution. Electrons are smaller than X-ray light you can obtain. So sometimes with well-ordered crystals, if resolution is an issue, micro-ID is method of choice. The disadvantage is that it requires a very small crystal, because we're limited to maximum 300 nanometers width, and with 200 kilovolts, it's probably roughly about 150 nanometers. But it's been very successful. So there are even commercial models of micro-ID instruments for chemical application, for chemistry, for small molecules. What we try to optimize our instrument is diffraction of proteins. Sometimes large crystals cannot be obtained, and then electron diffraction could be method of choice here. Cryoclam and feed milling. This is how samples can be milled. If you have a big specimen, you can mill a thin section. As I said, we have limitations of roughly about 300 nanometers, but the problem is as you tilt, because normally the feed milling is used for tomography, as you tilt your specimen, your cross-section becomes thicker. So in reality, for tomography, the limit is 150 nanometers for 300 kilo electron volts. And obviously, we offer a remote access. If people cannot come or choose not to come, all transmission electron microscopes can be accessed via can be accessed remotely. The only instruments which require users on sites are feed milling at the moment and like a cryo-lamp. So the access to Diamond is free on condition that people publish result. So in some cases, for example, if this is industrial partnership or people do not want to publish a result for some reason, we also have proprietary access. There is industrial wintered diamond. Obviously, it's not for free, but some funds can be obtained from LEAPS, from the European Synchrotron Organization, to cover costs. And also we have WINC and EBIC. It offers full service, so people just can come with a problem and get structure at the end, even some interpretation of the structure. It's also using all the capability we can offer, but also more advice. I think it's taken too long. Shall we? What would be the choice, to have a 5 minutes break or for me to cut that presentation, because we're pretty much halfway through?
It's up to you. We can make a short break for questions if you like. So let's ask people who. So please let's make a minute for questions. If you have any questions, please ask. You can raise hand or just it's not much people, so just ask.
Yeah, I cannot see participants, so please, if you can just give people.
I will help you if there are questions.
Mm-hmm. Thank you.
I will help you. Actually, when you were talking about initially about Diamond and university, what are relationship between Diamond and university, Oxford University? What is it? Belongs to university or what? It is private, you said, but
no, it's independent body. Although some people say that it's a backyard of Oxford University, which is not quite true. Okay. But yes, many people are employed at Oxford as well. But in general, we are independent body. So some people employed in other universities. For example, I-13 Beamline is a strong partnership with Manchester University. So obviously, university provide academic leadership, ideas, and Diamond is a facility to implement those ideas. And for example, if new technique is developed and it shows promise, just make it available to wider community.
Mikhailo, please. Microphone is.
Oh, yes. Yuriy, are you dealing with cryo-electron microscopy personally or not?
Yes, I'm an electron microscopist. I actually know much less about X-rays and synchrotron than I do know about electron microscope biological applications.
You're working with this Titan as well, yes?
Yes, I'm principal electron microscopy scientist. So we have 3 principal electron microscopy scientists. So we're different from Diamond in a way that we have sort of virtual beamlines. You see, we have plenty of instruments. Every of those instruments is run as a separate beamline. And that would make our infrastructure, basically our personnel, very convoluted. So, for example, we decided that There will be 3 people who do management of the resources, and we're interchangeable, which gives us flexibility. But yeah. So any questions you have, I can answer.
Because we are very interested in the studying by this cryo electron microscopy several kind of large complexes of micromolecules. And due to this, I ask you if it's possible to contact with you, or I write you a mail, and can I send you a letter about our interest?
Yes, you can send me a letter. I can also use different from English language, which is easier. Yeah, fine. I'll be happy to help, because that's what I'm doing for the last 22 years.
It is the main purpose of today's meeting.
Yeah, exactly. So the thing is, I left Ukraine like 22 years ago, and I currently have no contacts with science.
Більш того, ми з вами земляки, між іншим. Ви з Ніколаєва, я з Херсону.
Так, ми сусіди.
Okay, thank you.
Yeah.
Vlada, please.
Yeah, thank you, Yuriy, again for a fantastic presentation of the possibilities. I previous Solaris workshop, and now again enjoyed the big possibilities. Now I already selected the villages which can be interesting for us, for example, macromolecules crystallography, electron microscope, and so on. But I have, as Mihaila, also some formal questions already. How to start? So you described that it's possible to send samples. How it's possible? Which types of mail? I maybe say that I'm working in biophysical department, molecular biophysics department of Institute of Physics and Engineering in Kharkiv. So you studied in Odessa. Kharkiv is also, I suppose, almost the same brother city. So how we can start the cooperation? How it's possible to use such fantastic possibility for Ukrainian scientists? Maybe you will say this in other minutes of our meeting today. Would be very grateful. Thank you.
I'll cover it, just general information that obviously my purpose of this talk is to show possibilities. And actually, I mean, for many years I was really, how should I say, sad that there is such a marvelous instrument is pretty much free at the point of use, is not being exploited. And there are reasons for that, but I just try to make the border lower, because the issue is, in the past, people would need to know a lot of things about instruments, how to do experiments, and that all changes within last 10 years. The idea now is that people come with a question, and then a specialist can help answer it. Obviously, I cannot speak for every beamline, so my role would be to connect you and other people who come with a question to right people. You can also contact people directly through Diamond. If you see like particular beamline, so you'd like to get an answer, and you can contact directly. But yeah, I can also help in this respect. Yeah, so I think if you come with problems you would like to sort, I help you to connect with right people, and where I can help, I can help as well.
Yeah, my question is it free of charge, the possibility for Ukraine?
Diamond is free for charge for everyone. The issues are the cost of shipping of specimen and people travel cost. However, when people get inside the UK, normally travel inside the UK and accommodation for duration of experiment is operational costs. So Diamond reimburses this. So basically, Diamond will book a hotel for you and will reimburse you tickets to get, for example, from Heathrow to Diamond to do experiment. So what's reimbursed? Normally it's 5 days before the experiment, and duration of experiment and duration of experiment and 2 days after experiment. But this is, as I've been told, this is tunable parameters. So if your experiment requires you to stay longer, there could be other possibilities to extend it. So how it is covered, I'll touch on it later in presentation. Yeah, in general, it's much better, for example, than facility at university, because in all universities I worked, we actually had to pay for the equipment we use. Even the funny thing, like when in San Diego, there was national facility, which was free for anyone in the US, but we had to pay because we were located in the same building. Diamond is different, so everything is covered by the funding from government here.
Right. Yeah, I think it's usually there are many options, of course, and in order to understand it, it's the best way to try. Of course, you just send information to Yuriy, and he could your wishes, and he could suggest in which form it's better to realize. Either it's regular project you submit, which is the deadline is soon at the end of this month, right? And
yes.
And it could be a rapid option, and it could be collaboration, but okay. I see there is already 2 questions from Mikhailo Ivantsov in the chat. So first, is this institution offers any work for young researchers or just sample analysis?
Yes, there are schemes for young researchers. Please look at the Diamond vacancies. There are plenty of different fields. They're not necessarily even in science, because Diamond is a big facility. It's 700 employees at the moment and growing. So there's also some management and like outreach jobs. Yeah, the schemes for young researchers. So basically, if you would like full funding from Diamond, you need to look at what's available and vacancies.
Another question from Mikhailo was, will you share this presentation in a file form too, or only a record of your...
Yes, I can make file out of it, yes.
I will give you this link to it, to the places where advertisement I did and on the site.
Yeah, I'll convert it to PDF and maybe remove a couple of slides which too fresh to show.
Okay, if no more questions at the moment, then please, Yuriy, continue.
Yes, thank you. So, yeah, so I would like to continue with some EBIC highlights. So, despite we've been around for just about 10 years and really started operational user program in about 2015, we have quite a good record of user publications. So you see that our biggest is 70 publications per year, and the averages would be about 40-50 publications. About 1,000. Is it? I wanted to ask how many people, but is it possible to ask how many people? We currently employ about 20 people at EBIC, but these publications, it's not EBIC publications, user publications. I'm not sure if that includes publication of where staff was involved, but we use a facility. So if I set up experiment, I do not need to be on publication. It's not a collaboration. It's like a factory, if you see what I mean. So it's only in collaboration we get acknowledgement as a co-authorship. But our role is to actually provide people with place where they can come and collect data. So basically we try to adopt latest and greatest techniques, and we collaborate with many universities. You asked about universities. So yes, this is what we miss a little bit at the moment, because we mostly went remote, and quite often when users are on site, there's exchange of ideas. and not only between us, but between different university groups who are doing the same things and trying to address the same issue. So we tried to steer it up again a little bit. Yeah, so number of PDBs. So there's two data banks. You might know that protein data bank, where high resolution structure deposited, and electron microscopy data bank, where people deposit electron microscopy structure, and sometimes there could be one high resolution structure, but several structures from electron microscope showing dynamics, several structure of electron densities without this backbone arrangement, without atomic models docked. So there's larger number of structures in electron microscopy data bank. And this is examples from last half of year. You see quite high profile publications, and they cover very large spectrum of molecules. So Mikhail was asking about macromolecules. So here we go. This is complex 1, respiratory complex, membrane complex, very large. I think it exceeds a megadalton, I'm correct. And here we have ribosome, which is about 5 megadalton complex. Then on smaller scale is membrane attack complex, which is involved in immunity response in the cell. These are molecules which are existing as monomer, but once there's like invasion of bacteria of human body, for example, they activate and they create a pore inside the pierce a pore inside the bacteria and basically drain it out, so then the immune system can kill it. And then others are protein DNA interaction, also macromolecular interactions, big complexes like tencurase filaments in this case, and also bacteriophage complexes, which consist of 3 different types of subunits arranged in self-assembling complexes. And it's very interesting in terms, in part, that the quality of the structure has been involved in this cryo-EM. So some time ago, I was working on it and I got structure to about 8 angstrom and got some parts wrong because we just didn't have enough resolution to interpret it correctly. And now it's been collected, and if I remember correctly, 2.7 angstrom resolution. So everything crystal clear, problem solved. This is the power of evolution of methods over the last 10 years. The other project which I was directly involved and started during my time in Imperial College, and then I just helped to carry it on when I became Diamond employee. So it's chromatin remodeling, structure of chromatin remodeling complexes. Just to give you an overview, we all know about DNA and how important it is. So DNA basically stores information in the cell. But if you imagine that your computer start to talk to you about all data it has in it, that probably wouldn't be very helpful. So therefore, as genome gets more complicated, contains more genes, organisms had to come up with a way to control expression on that piece of information which is required at any particular time. And also to manage this very long stretches of DNA. For example, a human cell contains about 2.5 meters of DNA, which is compacted in very, very small space. And obviously, it needs to be ordered and managed and maintained. Then when cell divides, it needs to be compressed. When there is expression of particular genes, it needs to be decompressed. And the current hypothesis is that in eukaryotic cell and higher eukaryotes, multicellular organisms, majority of genome is actually silent. And only a small fraction of genome is open for transcription and to actually read and to execute instructions there. So you see this is like organization from DNA, double helix. Then the first level is the protein, which is called proteins which called histones. They are assembled into nucleosomes when they condense with DNA, and this so-called 11 nanometer bead, it's like bead on the string. If you look at the microscope, this 11 nanometer fiber we call it. And then the idea is that it's condensed into higher order structure, 30 nanometer fiber, which then condensed even more and more condensed material, like chromosomes when cell divides. So to remodel all this, yeah, so this is how the histone histone core look like. So there's the interesting thing about it. There are different types of histones, but they can only be assembled into particle when they all combined with DNA. The problem is histones control the access to DNA. So in the cell, there are different enzymes which modify histone tails, and the modification of these long histone tails would tell other enzymes, is this place of DNA which needs to be read, or is it place of DNA which needs to be silent? One of the causes, one of the most important causes of aggressive cancers is that this order is being lost. So the cell starts to express genes which shouldn't be expressed. It loses its ability to execute program of the whole body and try to multiply without control, leading to development of aggressive cancers. Therefore, proteins which work on this code and modify histones and modify structure of chromatin are very important in many in medicine and many like biological theories to understand how the whole thing works. I've been working on different chromatin remodeling complexes during my career, and the most recent one, which I worked on in Imperial College and then a little bit in Diamond, is SWER1 chromatin remodeler. The role of this complex is to replace H2A, H2A? Yeah, here, H2A histone with its different version, H2AZ, which leads to more compressed genome, and then there's another enzyme which does the opposite work. So it was shown that the combination of these two enzymes is crucial to maintain a healthy cell. If things go wrong, then the cell can evolve into more cancers. And as you see, this is very difficult to crystallize. It has more than 12 subunits. And even with cryo-EM, part of the complex is missing. You see on the 6 subunits is present here with 6 subunits because the rest is just too flexible to resolve with current methods in cryo-EM. But still, we can have a better understanding of how things work. So you see, we captured this complex with nucleosome, and you can see how the chromatin remodeling mechanism starts, because you see a motor protein of SWER1 capturing helices of DNA, distorting them and pulling them off from histone core, so the histone exchange can take place. So, as I said, one of the challenges which still remain is computational challenges. The computation is what holds back single particle analysis very often. The microscope allows much higher resolution than we achieve. So next, we're going back to EBIC and what we can offer users. I touched already on Chameleon. And one of the problems with Chameleon was that it requires really high concentration of a sample. So a typical concentration of a sample for cryo-EM, it's still possibly lower than for SACS experiment or crystallography. So one would require probably like, if you know your conditions, you would need like 20 to 50 microliters of sample at about, depends on protein, between 3 and 0.3 milligram per milliliter concentration. With Chameleon, it could double or triple. And that's possibly because the fact that something which hasn't been anticipated when cryo microscopy was born, people thought that you would have particles inside the thin layer of film, which you see, for example, like in the middle, or in the middle here. What you see, this is like image of atlas of a grid. It's 3 millimeter grid with central part, so it's square, it's not round. Then you see a square which has, in this case, I guess it's about 2 micron holes. And within these holes, there's a frozen thin layer of film, which normally goes between like 50 to 200 nanometers in thickness. Normally, it's about between 50 and 150 nanometers in thickness. And then then we take higher mag image from every hole, multiple images, so we can actually get this picture of particles. And the problem was with single particle in the past that most molecule would be attached to air-water interface. And this attachment, although it's bad for preferred orientation, and in worst case, it can actually denature parts of the protein or entire protein if your layer gets too thin, because then particle has been squeezed between 2 air-water interfaces. It also has concentrating effect, because if you have like roughly about 100 nanometer thick layer, and your particles then attach to very thin surface, and you concentrate your particles. In chameleon, because it's so fast, particles do not have this ability, and people saw that you need 2 to 3 times larger concentration, which for many biological experiments is an issue. So, and then when I was working on other object in Imperial, we adopted graphene oxide deposition procedure. It's known in many sources, but the trick was to solubilize it in detergent, which provides much better coverage of the grid surface, so it can get better coverage and very thin layer of graphene oxide. And that would require because now all your particles are attached, so your concentrating effect happens on contact. You can drop your concentration to about as low as 0.05 milligram per ml, like 100 to 50 micrograms per ml of protein. So when I joined Chameleon, I encouraged people to try, and now they adopted this procedure for chameleon grids. The chameleon grids are slightly different from conventional grids, so it wasn't tricky, but the result you see here, this beta-galactosidase sample, which is very nicely distributed, different orientations, and it goes to 2.7 ounce resolution and requires much less protein. It's even less than a conventional cryo-EM without support field would require. And also Diamond has helped SPT LabTech to develop some instrumentation to improve sample deposition, to keep it cold and control the temperature. So it's a collaboration which works for every party, I hope. So access to this instrument is pretty much to a beamline to a diamond, So if you would like to have access, you can apply, and then you get sample preparation and microscope time. The only thing is you need to justify why you, and that's another tool how to do it quickly. So this is our first commissioning visit on light acrylate microscope. You see a beautiful image of bacteria. This is a magnetic field sensitive bacteria. So in the middle, you see an array of particles, which are magnetosomes. This is a small, not sure what they're made of, but they're sensitive to magnetic field. They range only in one orientation and help bacteria to adopt this orientation, which for some reason is favorable for it for survival. So with this group, we did experiments on other beamlines like B24. What they missed there was throughput, because B24 soft X-ray beamline, I'll talk about it later, can give you a wide field of view, but it's limited in speed of screening. If you need a small object and a lot of data, then it's a longer experiment than B24 than it would be at EBIC. We have cryo-EM microscope, which allows fluorescence. So for this group, they wanted to study 6 different conditions and see how generation of magnetosome changes under these conditions. So they came for a 2-day visit. I think at this point, Yeah, they actually managed to screen them all 6 conditions. First with fluorescence, because the fluorescent label protein inside the cell, and then you can find the same. You can superimpose low resolution images. You can find the same object in electron microscope, and then collect either a single image or tomographic data to actually study the structure of the object and see inside. As with other equipment, we try to keep our equipment performing at best. We noticed that there is some problem. If experiment takes longer, you have some ice deposition. Then in collaboration with the Diamond technical department, we devised an equipment which would create a clean, dry environment around the specimen. So you see it's drastically removed these small specks, which is ice deposition is from atmospheric air deposited on the cold surface because it's all in cryo conditions. But we shouldn't want to have these problems. We still optimize the equipment to make it more user-friendly, but should be available for users soon. Then a bit unusual example. This is how experiment can be done. So in principle, to approve access to a beamline, we need preliminary results. So this is one of the proposals which people send to us, and they're looking here at devising a new carrier for drug delivery to cancer cells. So this is a cubosome, which are in fact lipid crystals, which can be filled with a drug. In this case, it was helinaine, which has shown some activity to kill some cancer cells. And they also wanted to direct it using magnetic particles. So it will be brought to a site where it needs to be most active, either magnetic particles or antibodies to attach to the cell. So it's the principle here. The problem is they had wealth of chemical material, but no confirmation that this would work with electron microscope. So I just suggested them that instead of, because I was pretty interested to see, to check this, I suggested that instead of putting it through normal access route, they can possibly have a collaboration and they can have a session to screen their conditions. And when the person came, asked how many conditions you have, and said 21. And I said, well, there's no way we can do 21 conditions in 2 days. So let's do 3. We optimized sample preparation because at this point I had no idea about this. Here is the images. So I had no idea about the specimen, how to approach it, what kind of grids we need. I said, let's do 3 conditions. Let's screen like 6 different types of grids. And then next day we put it on the microscope. We went through the grids. We checked where we have the best result. And then I also, in the meantime, I trained this person to use VitroBot freezing equipment independently. So this person just frozen the other conditions independently, and also it's not like extensive EM, but at this point, this is one of the examples when the most information would come from a still image, from a single still image. So it was like basic, like picture taking. So he sat down and took all the pictures required for the publication. The only tricky technical bit was then the question was where this metal particles, these very dense metal particles, are located. Because people were afraid that they're located on periphery, then they can be deposited in different parts of the body, which might be a problem. So we need to prove that they actually got inside. So I did the electron tomography of these particles. This is one of the tomograms. And in electron tomography, you can reconstruct a 3-dimensional volume, and we can go through slices, and you can see that this is like composite structure. You see that the metal particles are all in a line here, but if you go through the slices, you see that some of them actually touching surface, and some of them buried deep inside the cubosome. Right, the next part is electron diffraction. We have that ability, but we had commercial solution from Thermo Fisher Scientific, They adopted one of the older detector and made the CTD detector. So this principle, it's similar to CCD detector and it had very high noise level. So we got on loan Dectris, simple Dectris camera, and new camera from Dectris company in Switzerland, which we helped them to characterize and optimize. And also we optimized our workflow. So now this camera has been purchased by Research Complex at Harvard, and that will be available to Research Complex users and Diamond users via our partnership with Research Complex. Yeah, you see the stats here. You can use either like small crystals. Here it's in this case it was 1 by 1 by 0.2 micron large crystal, really small, and we had only 3 crystals to get the full data set to get 2.2 angstrom resolution. Previously, this protein was studied with X-file, which requires like thousands of crystals because you only can shot it once and you put it in a stream. So potentially, if you cannot get a large yield of your protein to create a huge number of crystals for free electron laser experiments, it can be done via electron diffraction with much better efficiency. And then some crystals, like protein crystals, they grow larger. And in this case, we need to mill it. So we can also have milling equipment. So in this case, we used, this is the beam, and then this is a small area we use for data collection in yellow. It's 1.4 microns. You see it still goes to about 2.2 angstrom resolution. And small molecules, it's with collaboration with, it's basically one group from other university would like to optimize the techniques that they got to us to see what they can achieve with our detector. And it was better than angstrom resolution information here. I think I'm not sure it did it go to 0.6 or 0.7, but it was sub-angstrom resolution. The interesting bit is that this equipment is complementary to what the VMXM beamline can offer. So sometimes people can bring it on the same carrier on cryo-EM grid, and then a decision can be made either to try micro-ED or VMXM, or try both and see which provides the best result. But also micro-ED would be available when synchrotron doesn't have beam. We prepare to upgrade Diamond to next generation synchrotron with 3.5 gigaelectron voltage. That would mean that for a few years, synchrotron will be, I think now the dark period planned from start to 2028, if government funding will be continued at current pace, but it could possibly get worse. I'm not sure it will get better, but that's the plan. So during that time, EBIC would be still operational because we do not need synchrotron radiation source for our microscopes. Next is B24 beamline. It's part of biological cryo-imaging suite, and that uses X-ray for tomography. The benefits is that it can look at larger objects. So for objects up to 10 microns can be used in B24. And that means that it can provide information about bigger context of experiments, which we can do at high resolution at EBIC. A good example is a study on cytopathy caused by SARS-CoV-2 virus. During work at EBIC, because EBIC was involved into a lot of work during the COVID-19 pandemic, and people noticed that when virus matures, it wrecks the cell, which might explain why people get so sick from this virus. The problem is we only look at very small area, and we needed to look at a wider perspective. What does it mean? And also scan more samples, and B24 helped to do that. So you see, this is a small image here, which shows what we see in electron microscope, and then in B24, you can look at pretty much same event, but you can trace it back to cell membrane and trace it back to the whole cell. More details in this paper. Another thing, I simply run out of time to make all the slides. I apologize for this, but I said it already before that B24 offers super resolution structured microscopy of cryo samples. So people can do, if there's fluorescent labels, people can do fluorescent microscopy first, and then put it either on the soft X-ray B24 beam line, or even take it to EBIC and study under electron microscope. In the way, it's similar to what we can offer with our cryo-FLAM, but at B24 it has high resolution. And the next one, we are going to have Biological Cryoimaging Day on CCPEM Spring Symposium at 25th of April. Unfortunately, some morning sessions are in person only, but otherwise everything else would be available online as well. So you still can register. Remote, like online, is free. They only ask for registration because they need to understand the numbers and how they proceed with organization. So people will talk about latest and greatest. We also will have some keynote speakers, and yeah, you can learn much more about things B24 and EBIC. And also, I think this year we have some representation from membrane protein lab. Right, let's move beyond the beam lines now. Also, we have facilities, XChem facilities. It's linked to beam lines and helps people to prepare for experiment. For example, if there is drug design, people would like to soak in different compounds, the crystals, or also, I mean, either you can come there for crystallization experiments. That's a question I need to get answer to. In principle, I think it should be possible, but some infrastructure can be arranged. And I'll talk about it, about potential problems there. So XChem facility was involved into drug discovery during a COVID pandemic, and it's a little known fact that this is a hugely successful international program, but they really relied heavily on supply of chemical from Enamine, which is a Ukrainian company, and they highly price actually the ability to produce the most complicated chemicals, which sometimes could be only theoretically described and then could be synthesized by this company. Sorry, I think I messed things up. So this XChem facility is pretty much for soaking, and crystallization facility is where you can come and try to crystallize. So in principle, this facility is available for people to come on a few days basis as a beamline, but for longer stays, it needs to be possibly funded by Instruct. And I already told in a few forums here that the problem is currently Ukraine is not part of Instruct. I don't know how to reach out to decision makers to say that maybe something needs to be done about this, because this is an infrastructure grant. And it basically gives you access. It's not linked at the moment to any expiration dates as other techniques. It relies on country paying some money into it, but I think given the circumstances, other arrangement can be thought, but someone needs to start talking about it on state level, I guess. And then XFEL hub, that's the connection. If your experiment requires a free electron laser, so obviously one can apply through other directly or other institutes, but at Diamond we have, you can apply here, and then your samples will be shipped to XFEL, and you do free electron laser diffraction taking. But that sometimes requires very high yields of small crystals to get data. Membrane protein laboratory is hugely successful project run from 2008. And this is fine-tuned for people to come into member protein studies. It's not only structural studies. As you see, there's also other techniques available to characterize your sample and obtain structures. It was for years, it was using crystallography, but now they going into electron microscopy business. The many structures which obtained, it's in UK and also from other places in the world. The applications, it's also, one can do a planned application and rapid access. With rapid access, one thing that the justification needs to be done why this urgent request is required, because it's like non-standard request. The things it can offer is screening your sample, preparation, screening, for example, if you go for microscopy, there could be negative stain microscopy, which EBIC doesn't offer, but because NPL is in research complex, they have access to lower end gel microscope, which can use negative stain. It's stained with, normally it's heavy metal deposition, heavy metal salt deposition, which creates this nice contrast. You see your particles much better. You don't need to freeze it. You can see that from temperature, but then also that basically gives you fast feedback on quality of your sample. You can do it within hours as opposed to probably a day with cryo-EM, because the time for sample preparation takes quite some time. The microscope itself. The microscope itself probably isn't that long anymore, but sample preparation for cryo-EM is a bit longer. It's hours rather than minutes. And yeah, also they have mass spectrometer, dynamic light scattering to see properties and if sample aggregates and forms bigger complexes. And how people would find what technique is right. So as I already said before, there's EBIC web page, sorry, EBIC Diamond web page. One can check different science groups, different instruments. There's also a very nice array of studies and case studies in industrial liaison industrial liaison part. So this fine-tuned for industry, but in principle, this science is done with Diamond, so one can read and have ideas how what beamlines can be necessary, what kind of problems can be solved at Diamond. Submission of proposals is applied for beamline. So if you have a well-formulated proposal, you can go to this and read what type of access you need. So normally, most beamlines would operate with standard access. That means that the call for proposals is twice a year, and then it's going on technical review by Diamond staff. And we say, is it feasible or not? Does user provide significant amount of evidence that experiment will be successful? This, however, is different for every beamline. And this is possibly where people need to inquire to ask what would be required to get standard access. So for example, for EBIC, if it is electron microscopy, we would like to see if it is like single particle, we would like to see successful cryo-EM grid, 2D classes to show that this is a good quality sample, and sometimes low resolution 3D model, which already requires access to facilities. Unfortunately for us, to get access, I mean, some EM should be done. And that's possibly the majority of project. If I'm correct, probably there's no such EM facilities in Ukraine at this point. Majority of project probably should start as collaboration, I believe, with EBIC or other facilities to get access to the microscope, to get at least negative stain pictures to say that this is a good quality sample, and we can achieve a good result. For other beamlines, for example, P24, they say that they're happy to have this optimization, but they would need confirmation that samples can be cultured. Normally, these samples are cells. So they would like confirmation that samples could be either deposited on AM grid or be cultured on AM grid. And then from there, they can help to develop how to freeze. They have a plunge freezing device where you can vitrify your specimen and then take it from there, learn the technique, and do the experiment. So for other beamlines could be other criteria. They should be described for every beamline. If you go to their pages, they normally describe what criteria are required for successful application. For rapid access, the reason should be given why this experiment is urgent. For example, if figures of information is required for publication, or there is a competitive project which you would like to compete with, and you need your result quickly. Some other reasons. Also, some preliminary data are required. And some beamlines, like for example, IPSIC says that they're very happy to answer questions before the proposal is actually written. To sometimes voting is all it takes to make unsuccessful proposal a successful proposal. So if you ask how to vote it. And there's also a joint scheme for ISIS neutron immune source. I wasn't able to get any slides on joint research, although I know that there is such joint research between EPIC and Diamond. But one way people use ISIS neutron immune source is sometimes to mimic environmental changes, because in principle, you can just make time run faster with this type of radiation. And if you would like to see how your material would age over time, or how your cell, how your environment will change for biological materials, that can be combined with measurements in Diamond and implemented at ISIS. And then the other thing is that we have quite a big success rate. So if proposal is well written, you have about 60% chance that it will go through. And I wish it was the case for some my grant application, which unfortunately have much lower success rate. But the other thing is that many sessions are remote. So it's obviously not all beamlines. Most beamlines probably have some type of remote access, but not experiments can be run remotely. But we see that at least half of our experiments are remote, and for many biological applications, like I know that macromolecular crystallography and EBIC, almost everything can be done remotely. With B24, you can do it remotely, but they prefer to be in person because the nature of their tomography at the moment, the image gets, when you do tomography, you see image straight away, at least in this kind of beamline. And sometimes you need to take decisions, and the quality of internet link is not sufficient for you to see the fine detail. Diamond is working to improve this, but at the moment, this is why coming on site could be better, at least for B24. Right, so the access. So as I said, there are two ways, well, different, sorry, apologize, I went too fast. Another big thing is bag access. Majority beamline don't have it, and EBIC we have a bag access. So, and bag access is an interesting access, because with rapid access, for example, and standard access, you need sufficient proof that your experiment can be done. With bag access is which is allocation of chunk of time at Diamond. So you just need to demonstrate that your science is sound, and at least for some of your experiment, you need to demonstrate that cryo-EM can work for them. but then you just allocate a chunk of time. You use microscope during this time. Obviously, all your samples need to be validated from the user administration system, but people can put samples which were never seen by cryo, which possibly I shouldn't say here because this leads sometimes to bad experiments. But yeah, this is part of the deal. Nobody can.
What is BAG? What is BAG?
The BAG is block allocation group.
Block.
Yes, block allocation group at EBIC, it's either 80 or 90% of time. We only have about 10 to 15% time allocated to rapid access. We don't have standard access. Rapid access, every experiment is a single, every access is a single experiment. It might be like if it gets awarded like several sessions, but it normally like 1 to up to 5 sessions. And that's it. Then after your time is over, you need to apply again.
Okay, for me it sounds complicated, but you should learn it only by experience.
No, it's important bit. So if you need to spend more time, I'm happy to spend time on this because the problem is you cannot have rapid and BAG access. So we turned down some rapid proposals because people have BAG time. So BAG is, it cannot be done by one person. But for example, if there are a few PIs, a few principal investigators, an institute or a few institutes who decide to create a BAG, they can say that, okay, so this is our proposal. It's a joint proposal always. It should go across different groups and across sometimes cross institutes. And the beauty of it that you can provide just some examples of science you would like to do. And based on these examples, you allocated time. But you pretty much can run experiments. But
this should be a successful example realized already before in this your lab, or?
Yeah, some examples in this BAG can be successful. But for example, Ukraine can create a BAG with Poland, which has EM facility and has where people can get some EM data out. Possibly the capacity will not allow for like 5-6 groups, right? But it will be sufficient to get data, evidence the project can be successful to apply it to Diamond as a BAG. And then all these 5-6 groups can send samples to Diamond to collect data.
Okay. Yeah.
Okay, so yeah, so this is basically different aspects. So, but I think for now, if people interested, let's discuss the science, and then we can find the way. It's different. It's intricate. This is why I felt this talk is necessary. I felt when I first started, I came to Diamond first as a user, and I was, let's say, I was really angry because for me all this system was seeming to be too complicated, annoying. All these emails you get, you need to tick all the boxes, go through all these checks. But once you learn the system, you understand that it gives you capacity to do really, really great things. So, and then you start to appreciate that maybe there's complexity for a reason. Right. So then we move into iNext scheme. So it gives access to users to several beamlines: B24, B24, B21, B24 EBIC, VMXI, beamlines, VMXM, XCAM lab for fragment screening. I think you can also get access to member proton lab, although they preferred option is instruct. But iNext Discover also can be used as far as I understand. And Ukraine can be part of it. You can apply here. I don't have information how other beamlines treated. In EBIC, this is treated as rapid access. The catch here is that sometimes you don't really need as much information here, as much preliminary data as you would need to get rapid access. And you don't really need to say that this experiment is urgent because competition or because you need to write a review. So INEX Discovery reviews it based on scientific content. So once refers to technical review. So if we're happy that this is feasible, for example, you have like even negative stain or simple cryo-EM image with your particles, and you show examples of some particles or some classes. And this is our technical assessment. So I can see that basically, this is not as elaborated as it is for other project run as rapid proposals. And it's allocated as rapid proposal. So you get this time, you need to spend this money because it only runs until I think June next year. So Diamond is really keen to get this moving, and as much time as possible will be allocated to INEX Discovery proposals. But it's a dual application. First goes to INEX Discovery, then it goes to us, and then you get time on the microscope. The INEX Discovery, the other beauty of it, it will cover everything. So, for example, if the majority of experiments Diamond cannot cover international travel, it will cover it for not Diamond. Basically, Diamond will cover and then seek reimbursement from INEX Discovery. Eric Instruct, I already mentioned that this is something which gives access for months. It would be nice to get this moving. And for industry, the LIPS Innovation Fund, it's for small companies and enterprises. It needs to be applied. It's a European grant. It's access to European synchrotrons. Diamond is part of it, and happy to give time allocation if grant is successful. And this is pretty much end of my talk. I really thank you for your patience. It's a long talk. I would like to thank everyone who helped me to prepare the slides. And thank you for your attention. I'm happy to address your questions now.
Yeah, thank you, Yuri. It's a great effort to make such a talk.
Yeah.
So please, questions. I saw the question on the chat, but that was about software. Just I think it appeared when you just before when you started to talk about machine learning stuff, and that was about software. Whether you're using, what software are you using to in order to obtain 3D models of proteins, DNA, etc.
Right. So for single particle experiments, we're using Reliant software. So we have a lot of software installed. So historically, I sometimes use software which is about like 20, 15 years old, just to get some unusual things which are not implemented in recent packages. But for on-the-fly processing and standard is Reliant software. We currently use Reliant 4, or their versions are available. The other software which we do not offer for users, but it's installed on site, is CryoSpark. So the benefit of Reliant CryoSpark is new generation software which uses maximum likelihood and stochastic gradient descent techniques for cryo-EM to get quicker, to get more efficient models. This wasn't available in earlier generation, which was mostly based on statistical methods like classical statistics, like signal processing methods. They have a bit of both because we cannot really walk away from. I wonder if we, for some reason, I just removed the slide with conclusions. Yeah, so you can use a little bit of both. Unfortunately, for users to get access to CryoSpark, they need to be affiliated with Diamond. So I think Yeah, but CryoSpark can be installed. It's free for academic users. It doesn't require very big clusters because Reliant is really computational hungry. It requires CPU and GPU clusters to be efficient. CryoSpark, I think now it can be run in clusters, but in principle, it requires standalone computer with powerful GPU. You can check what's exactly required. We also have partnership with CCPEM. This is collaborative computational projects in electron microscopy. One of the main offices is based in the research complex. So they help us to set up and run Reliant pipelines. And they also develop other tools beyond the analysis and getting 3D structure. For example, how to interpret your 3D structure, how to automatically dock atomic coordinates, how to build your model, because it's very well developed in X-ray crystallography. It was not developed well for EM, but now more and more tools are developing, and CCPEM is leading development of such tools. Yeah, this is pretty much in the particles part. Tomography, we're experimenting with many things. The main is iMode. This is like classical software. The other software is available, but less used in our place. At the moment, actually building a pipeline around a new software which is called Atomo. And Reliant 4 also got capability to do subtomogram averaging to get structure of molecules in situ. Unfortunately, I didn't have time to put together a slide, but it's really spectacular if you can identify multiple copies of your protein or your structure, like filaments inside the cell, and you can extract them and average out. You can actually build the structure in cellular context. And I think if you go to BCI meeting and CCPM meeting, Spring Symposium, you'll see a lot of examples in that because it's new and fast developing field.
Okay, thank you. So if you have questions, just start to ask or raise a hand. Okay, Vlada, please.
Yeah, Yuri, again, thank you very much. You answered on some questions which I had before. Actually, when Alexander sent the information about the deadlines for application of some project, I tried to do and also realized that maybe it needs cooperation group in Diamond facility. Yes, because they asked federal ID or something like this. So now I realize that it's the best way is to establish the cooperation, and maybe I will write to you letter with the tasks which we have, and maybe you will give some advice with whom we can establish such a cooperation.
We can have a meeting and discuss
Okay.
things because you don't
Thank you.
I'm writing things, and just drop me an email. We'll discuss
Okay.
what you need, and
thank you very much.
we'll take it from there.
Yeah, it's clear. It's very understandable for me, especially for I'm from different area, of course, and never measured something like biological. Yeah, but I quite understand the requirements that you need. I understand that you need to demonstrate in your proposal. You need to demonstrate some abilities that your samples could be measured, and it is a nice idea to have some images of from some microscopies. But from my side, Mikhailo, you wanted to ask something? Or? Because
are you missing about socks? You should have on the demand socks.
Yes, we have socks. Vitaly, can you comment on that?
No, because SACS, it's also some additional tools for molecular structure and okay.
Yeah, no, I'm just asking my colleague Vitaly Mikhailuk, who is in MX Village, and
Yes, we do have dedicated few beam lines to measure SOX. Standard experiment can be arranged there. А можу я по-українськи відповісти?
Зрозуміло, що можеш.
Буде ще краще.
Звичайно, ми маємо САКС, біосакс, спеціальні бімлайни, і вони роблять весь набір експериментів, який може вас зацікавити. Знову ж таки, звертайтесь, і це, що треба, ми допоможемо. От, з цією інформацією, сказати, до кого звернутися, до якої людини, це все можна зробити.
Це ви маєте на увазі на діаманті, да?
На діаманті, так,
Ага,
так.
добре.
У мене був проект collaboration з chromatin remodeling протеїнами. Вони, там був дуже важкий проект. На жаль, вони звернулися вже досить пізно, коли залишилося півроку до кінця аспірантури. І студент закінчив аспірантуру, захистився, це дуже гарно, але найкращий результат, який вона отримала, був саме САКС. Ми змогли отримати достатньо гарний результат з мікроскопією, тому що technical problems. Але САКС так, ми маємо.
Дякую. Розуміло.
Можна я скажу ще два слова? От, на жаль, час цієї поширення інформації не дуже вдало для нас вийшов, тому що ми вже, це кінець фактично періоду, коли приймаються аплікації, а наступні аплікації на проекти це через тиждень фактично дедлайн, а наступна аплікація буде через півроку. Звичайно, це втрата півроку. Але що я хотів би, хто хоче, хто має якийсь інтерес, не тяніть з цим і починайте зразу, тому що з'являються такі можливості, наприклад, в MX Village, що цей експеримент можна зробити фактично буквально в режимі реального часу. Якщо є цікавий експеримент, можна написати що називається Rapid Proposal, і його швидко розглядають. Якщо цікаве щось, то приймають і роблять приміряння. Це стосується і MX, і біосакс експериментів. Тобто головне не тратити час. Якщо є інтерес, робити зразу, як з'являється таке конкретне завдання.
Ні, гарний привід, якщо ви подали статтю в Science чи Nature, і вам там пишуть, що знаєте, це гарна стаття, але от тут, якщо б у вас трошки збільшили роздільну здатність, то ми б її опублікували. І тоді ви посилаєте...
Ви прекрасно розумієте, це є, можна сказати, гарантовано вже тоді, якщо ви прийдете з таким рецензією рев'ювера, це, звичайно, практично гарантовано.
Це без черги, це без черги.
Так, я не говорю про такі екстраординарні випадки.
Взагалі, справді в мене, я нічого не розумію про біологічні зразки, мається на увазі.
Ви добре розумієте в магнітних матеріалах.
Але в мене питання було, справді, а що це таке, оце графеноксид, графеноксид, і він такий, звідки, як це формується така маленька структура, там скількись ангстрем, 2,7 ангстрем у вас було?
Це один молекулярний, мені важко українською, тому що я вчив всю термінологію назад. Слой, так. Я буду... Монолеєр, так. Це, проблема в чому? Що люди б хотіли б мати графен. Але з графеном проблема в тому, що він гідрофобний. І наші зразки, вони в водному розчині, тому дуже важко на графен їх нанести. І зробити сам layer of графен. Тому зараз вже навчилися люди. У нас є людина, яка робить дуже гарний графен. Ми отримали, якщо одну з найкращих роздільнодатностей саме завдяки графену. Але графеноксид, він може бути як колоїдний розчин, і ви просто берете каплю, наносите на мікроскопічну сітку, і якщо підібрати параметри правильно, то у вас там буде, ну, то, що я заміряв, там було від 1 там 3 слоя графеноксида. Для нас це не має значення, хоча деякі люди кажуть, він не проводить електрони так дуже гарно, як графен, тому що там є ідеальною arrangement of molecules, там Oxygen є. Але якщо нам треба якийсь супорт, який майже ми не бачимо.
Ви його просто використовуєте для покриття, да? Чи
так, просто щоб
як графітизація, да? Чи покриття металами, а це графеноксидом?
Ні, ми просто де сітка, у нас є металева сітка, так? На цю металеву сітку нанесений слой аморфного карбону.
Ну, це щоб зразки не заряджалися, я маю на увазі.
Ні, це щоб нанести на щось зразки, тому що наш зразок – це молекули. Повинен бути якийсь носій, правильно? Цей носій наноситься в нашому випадку, це сітка, це 3 мм або мідна, або якась металева сітка, але це повинен бути метал, який не магнетик, тому що одного разу експеримент люди не змогли робити, вони там був як електронний мікроскоп там магнитний. І на цю сітку нанесено, це вже комерційний. Можна зробити самому таку сітку. Ну як, ти купляєш металеву сітку, а на неї наносиш якийсь супорт фільм.
Я просто масштаби не розумію. Так, а який у неї розмір, ви кажете?
3 мм.
Мікрони?
Діаметр 3 мм.
А відстань між цими?
Вони бувають різні. Я не пам'ятаю dimensions, але є там десятки мікронів. Давайте зараз на презентації.
Ні, я просто так, а що у вас був за розмір 2,7 ангстрема? Це розмір чого?
Це роздільна здатність структури, яку ми отримали.
Роздільна здатність структури.
Так. Це роздільна здатність біологічної структури, яку ми отримали. Що ми можемо бачити атоми, якщо ми використовуємо microED. Це не для біологічних матеріалів.
А який розмір оцих, як ви казали, це якісь магнітні ланцюжки у цих, як вони, бактерії, да? У бактеріях магнітні ланцюжки, а там який розмір цікавий?
Майже вся ця половина цієї бактерії вписується в діаметр 1,2 мікрона. Десь 1 мікрон. Це бактерія десь завдовжки, десь 2 мікрони.
Угу. А звідки ви знаєте, що вони магнітні, ці ланцюжки?
Це біологія. Це вже зрозуміло.
Просто
це вже об'єкт, звісний, так.
Просто ви їх. Взагалі, от як, я розумію, що розшифровка таких молекул, по-перше, дифракція, зрозуміло, що якщо це там якась ДНК, то там періодична структура, воно дає дифракцію, да? Тому і орієнтацію, ви бачите тоді дифракцію, орієнтацію. Але ви бачите також зображення, і зображення ви намагаєтесь ідентифікувати за допомогою методів машинного навчання, зокрема, да? Чи правильно?
Зображення, нам треба статистика, тому що електрони, вони дуже, електрони просто спалюють зв'язок. І тому у нас дуже низький signal-to-noise ratio. Статистика необхідна нам, щоб щось побачити там. Якщо ви пам'ятаєте мій слайд, коли я показував частинки і 2D class averages, то ви бачите, що майже нічого, це сіре на сірому.
Оце сіре на сірому, це зображення
так, це
ваше,
single
а потім
particles.
ви вже його
Так.
витягаєте звідси сіре на сірому. Ну, я
Так.
так і зрозумів.
З нього ми отримуємо сигнали і трьохмірну модель.
Ну, тут великий простір для, ну от я в принципі тому і питав, що зрозуміло, що тут великий простір для дата процесинг, да, дата аналізис, як його відповідно, як з зображень витягати, але я також розумію, що в більшості випадків це вже зроблено у цьому софті, який є у цих приладах, да, і питання, чи залишається для дослідника, а і наскільки це важливо для дата-аналізу?
Це насправді проблема, тому що проблема з біологічною електронною мікроскопією це не лімітація інструментів. Мікроскоп може, ну, зараз є дискусії, що треба зробити, тому що теоретично може отримувати результатність менш ніж 1 ангстрем. Але фактично люди зараз топчуться біля десь 1,4, 1,2 ангстрема. Це максимальна результатність. Але більшість зразків, воно не досягає результатності, тому що ми не можемо їх прорахувати. Це computation проблема, а не інструментальна проблема на сьогоднішній момент. Ну, насправді, Diamond дає можливість отримувати результати. Якщо є серйозні наміри робити обробку даних, це Diamond не гарантує. Тому треба подумати, де ця обробка даних може бути зроблена. І може в Україні може щось буде створено або якийсь спосіб. Є зараз інтернаціональні cloud services або інтернаціональні facilities, де люди можуть запросити доступ і зробити обробку даних теж. Про це тут треба подумати, тому що деякі проекти, у нас зараз проект, які вже, пару проєктів, які вже там ковід був, все, але вони вже кілька років висять. І ми намагаємося їх доробити, але проблема зараз, що у нас великі масиви даних, і просто їх нема де хранити, тому що Diamond, це як revolving door. Дані записані, оброблені, наступні дані записуються, оброблюються, але всі дані йдуть на архівацію, і вони там на сьогоднішній день, вони там назавжди. Скільки назавжди буде длитися, ми ще не знаємо.
Юрій, я тебе перерву. Є європейський грант по суперкомп'ютеру, по гріду європейському, і там можна якось через членів Європи туди всунутися і порахувати.
Так. Це багато.
Є такий грант, він, кажеться, до 24-го року працює.
Ну, так, а взагалі, що open database, вони ж багато таких, певно, яких можливостей, чи, да, це ж саме для зберігання такого типу даних, да, ну, я розумію.
Не зберігання, а обрахування, треба обрахувати, треба, щоб
крім того, що тримати
був
їх
софтвер для того, щоб це обрахувати.
Добре. Це таке.
Можна технічне, Саша, технічне питання хотів спитати.
Паша, я якраз хотів спитати, Геомет тут з'явився, давай.
А як кріоелектронна мікроскопія, чи застосовувалась вона в ваших руках до взаємодій ліпідних мембран з білковими молекулами? Чи презервуються такі зразки і можна їх досліджувати чи ні?
Ну, так, саме це, що робить Membrane Protein Lab. Це їх, я можу показати.
Ну, Павло доєднався, я думаю, пізніше просто,
Там же
я наче був, але я прослухав.
Такий, да.
Зараз я знайду. Це все мембранні протеїни. Це Ось, ну, дивіться, це ви можете зробити lipid vesicles. Як вони називають українською, я просто не знаю, вибачте.
Ні, дивіться, я це теж слухав, це мені зрозуміло. Але якщо хочу взяти свій шматок мембрани, не свій, а об'єкта.
Маю на увазі печеклам, так?
Тобто я хочу, наприклад, там мікрон квадратної мембрани і там декілька десятків білків однакових, які певним чином асоційовані з поверхнею.
Якщо ви можете це заморозити, то ми це побачимо.
Добре, тобто це можливо, да?
Так? Так, це можливо. Ми можемо подумати, ну, дивіться, я можу тут допомогти, тому що я, в принципі, розпочав в Одесі з біофілічної лабораторії, і ми займалися трансмембранним транспортом, регуляцією гомеостазу різних іонів і генерацією трансмембранних потенціалів. І потім моє PhD – це саме транспортери, це VA type of ATPases, F-ATP synthase. І, в принципі, це біофізична хімія, тому в мене є експірієнс в цьому Diamond, в цьому зацікавлений. І так, у нас є метод.
Ну так, тоді давайте не затримувати всіх. Якщо можливо, я з вами, може, тоді зв'яжусь і поговорю напряму. Дуже дякую.
Да, дуже якраз цікаво, той слайд, що показували, якраз таке саме ось з візикулами, з ліпосомами ми досліджуємо лікарські препарати, якраз взаємодію такі, які є мембранотропні, звуться там дікамітоксин та інші. Так що дійсно, якби перехрестя є багато. Дуже цікавий той слайд, останній якраз.
Ну, я розумію, що я можу, Паша, переслати ваш... Ой, Паша, не Паша, Паша, я можу по мейл Юрія переслати так, усім зацікавленим, да, Юрій? І, відповідно, значить...
І Михайло, і Віталія теж.
Ну, так.
Віталій.
Так.
Віталій.
Да, дякую.
Добре.
А можливо, і презентацію нам розійшліть. Нас так то багато.
Презентацію я дам, мені просто треба її конвертувати в PDF,
Так,
ставити.
і презентацію, і відео, от, відповідно, значить, Юрій скаже, які прибрати слайди. Ну, верніше, слайди він сам прибере з презентації, а з відео який?
Там один слайд, який, він дуже, це буквально було роблено тиждень тому, і ми просто... Дуже гарний сайенс, ми попросили цей слайд, але вони не хотіли його комусь показувати.
Добре, дякую.
І може ще одне маленьке питання. Як в зрівнянні з Х-кристалографією, так, яка, мені здається, теж має роздільні здатності близькі до 2 ангстрем, тобто чим таки краще кріо-ІМ в зрівнянні з X-ray кристалографії?
Я тобі відповім, кристала не потрібно.
Ні, ну це зрозуміло, але якщо він є.
Зрозуміло. Ти попробуй кристал потримати, а потім зрозумієш.
Не пробував.
Отож.
Не пробував.
Є плюси, є мінуси. Тому що так, кристал не потрібно, але край-ІМ на сьогоднішній день досить повільно. Наприклад, якщо ви хочете подивитися, отримати високу роздільну здатність і подивитися різні там драгс чи ліганди, це буде досить повільно. У кристалографії, якщо у вас є кристал, ви можете прогнати всі ліганди досить швидко, це автоматизовано. Ми зараз тільки починаємо європейський грант, щоб оптимізувати це для мікроскопії. Це буде девелопмент методів, девелопмент сам робототехніки, сам computational technologies, artificial intelligence, багато чого. І, в принципі, може через років 5-10 електронна мікроскопія нас дожене в цьому кристалографію. Але так, ну, у нас як? Деякі люди називають наші математичні методи crystallization in silico. Що так, вам не треба кристал, але щоб обробити це все, вам треба примірно той же час, що отримати кристал. Але бувають Бувають з цього, це не правило, тому що я коли прийшов до Imperial College, то тоді мікроскопія ще не була настільки сильна, і мій шеф робив експерименти кристалографією. Але проблема була в тому, що деякі речі не можна отримати в великих кількостях. Наприклад, його основний проект був DNA helicases. Тому там є вилка DNA, і одна частина цієї хелікази, вона стає flexible. І це ми змогли вирішити за рахунок мікроскопії.
Можна я додам 5 копійок з своєї? Тому що це є взагалі-то взаємодоповнюючі методи, тому що є речі, які кристалографія робить набагато краще. Електронна мікроскопія, кріоелектронна мікроскопія, вона добра для великих молекул. Маленькі молекули біологічні, вона практично не дає вам ніякої інформації. Це можна зробити з кристалографії. Це набагато більше ти отримаєш інформацію. Так, це вірне зауваження стосовно додаткового кроку, який потрібно пройти для того, щоб здійснити експеримент на рентгенівської дифракції кристал, власне. Це так, але знову ж таки ми, роблячи цей експеримент, є можливість, допустимо, зробити ідентифікацію якихось іонів, які зв'язуються з твоїм протеїном. Встановити, якщо є, скажімо, якісь ліганди зв'язуються, поєднуються з ним, можна зробити їх кращу орієнтацію. Так що тут не треба жодне відкидати. А стосовно труднощів вирощення кристалу, то я панові Михайлові скажу, що як у вас є хороший протеїн і у вас є труднощі з вирощенням кристалу, то ви зверніться до нас, ми можемо це попробувати, виростити.
Так, до речі, це в принципі application for crystallization facility.
У вас вже є роботи. Раніше, коли не було роботів, то ми були на першому місці, тому що ми самі вирощували. А зараз, коли є роботи, то не треба рук. Все зрозуміло. Ну, Паша, кажеться, Віталій, я теж свою копійку добавлю. Зараз все більше і більше нас цікавлять в білках рухливі частини, які можуть приймати участь і в тих функціях, і в тих. Тому взаємодоповнююча мікроскопія дає можливість оці якраз домени дуже рухливі, які не видно в кристалах, X-реї не видно їх, вони там дуже рухаються і не видно. Електронна мікроскопія дає подивитися. Наприклад, в рибосомі людської є там такі длинні шлейфи. Як електронна мікроскопія точно б їх не побачила, в смислі це X-ray, а електронна мікроскопія бачить дуже длинні такі частинки білка. Оце така річ, да. А нам все зараз цікавіше і цікавіше такі лун-мун, це такі, як вони кажуть, інші функції у білків ми виявляємо, яких раніше ми і не виявляли. Оце дуже важливо.
Зрозуміло.
Дякую, ви сумували дуже гарно. В принципі, я думаю, що те, що Віталій сказав, що так, ми маємо проблеми з маленькими протеїнами. Зараз в мене є ще один проєкт, де ми намагаємося це покращити, але так, якщо протеїн менше, ніж 70 кілодальтон, це не неможливо, але це технічно важко стає. А 30 кілодальтонів вважається взагалі на зараз границя, за якою електронна мікроскопія просто не вистачить averaging технік. Люди намагаються це спростувати, але подивимось, теоретично там не виникають проблеми. Але їх немає у кристалографії.
Добре, а я ще хотів спитати, Паша точно по початку не бачив, мені здавалося, а там слайд там це у Віварі там ви показували є, да? Значить,
так, є Віварі, так.
І наскільки у вас є взаємодія, там, грубо кажучи, можна не зі своїми мишами приїжджати на синхротрон?
Ви повинні розуміти, що тут це буде дуже дорого, і вам прийдеться заповняти дуже багато.
Це важка історія.
Ні, це можливо. Я спитаю,
Скоріше навпаки відбувається.
Моя колега з ними хоче співпрацювати. Вона, на жаль, я теж не встиг зробити слайд. Вона вирощує нейрони на сітці, і вона дивиться на взаємодію між нейронами, як нейротрансмітер проходить крізь щілину між нейронами, але зараз вони не працюють, в них там якась реструктуризація, але в принципі так, це можливо. Ну як, це заповнення всіх цих паперів, це просто для мене це новинка, але все це працює, тому що тут дуже багато етичних моментів. Ви повинні показати, що, перше, вам необхідно цей експеримент саме на живій тварині. Повинні показати, що ваш експеримент спланований так, що ви будете мати не більше, ніж треба тварин, і показати, що з іншого боку, що ви будете мати достатньо тварин, щоб зробити ту науку, яка вам треба. І що з тими тваринами буде етичне походження і все інше. Ось, в мене немає просто у цьому. Я знаю, що треба, тому що це пункти, які...
А взагалі проводяться експерименти на живих мишах на синхротроні? На синхротроні, на рентген.
Я не впевнений, що у нас проводяться на живих мишах, але на органах...
На органах, а на живих не буває. Це...
Я не думаю, у нас немає такої категорії. Ми категорія 2, це, я думаю, категорія 3 вже. Особливо, якщо це генетично модифікований організм. Я не певний, як це. Я можу спитати. Я знаю, що через дорогу у нас Розенфранк інститут. Я маю розуміння, що у них є така можливість.
Так, добре. Значить, чи є ще останнє питання якесь у когось може? Тому що я бачу, що всі, певно, втомилися. Добре, я дуже дякую, вдячний Юрію, звичайно, за великий цей і доповідь, і усі, значить, Віталію також, всім, хто взяв участь, ми поширимо і, будь ласка, питання до Юрія, Віталія задавайте, вони будуть, як бачите, раді відповісти. Дякуємо вам дуже.
Дякую вам.
Дуже дякую.
Буду радий відповісти на ваші питання, емейли.
Майте гарні вихідні.
Щастить, на краще.